So our next speaker is Laura Fraticelli from Technical University Munich, who will talk to us about the energetic profiles and distribution of neurons and glia in the human brain.
Hi everybody, thank you very much for the introduction. Yeah, my name is Laura and I do my PhD with Valentin Riedl in the Neuroenergetics lab. And there we really focus on the energy metabolism of the human brain. So I'm very happy to share some part of my PhD project today with you. First of all, I have no disclosures, other than maybe the fact that I'm quite nervous and I can really feel the adrenaline like pumping through my body and making sure that my brain gets all the energy it needs right now to get me through this moment. And while our brains are like relatively small in size, so they represent only 2% of our body weight, they consume over 20% of our whole energy budget. And the main energy substrate in the brain is glucose. And glucose we can measure in the human brain with FDG PET. So FDG PET really allows us to measure the glucose consumption on a voxel level. And so the origin of this like signal that we measure with FDG PET actually relies on a bunch of different cells. So if we consider a voxel of like 1 millimeter cubic voxel, we actually have over 57,000 cells in there. And while there's many different cell types, majority are really neurons and astrocytes. So these two cell types actually possess very different metabolic profiles. So generally we think that neurons consume really majority of the glucose in the brain to really fuel their very costly signaling processes. So therefore they mainly metabolize glucose through a process called oxidative phosphorylation, and and this really allows them to generate large amounts of energy in the form of ATP. On the other hand, astrocytes are known to be more like supportive cells and they don't really rely on this high energy production, but are thought to be more like glycolytic. So they mainly consume energy through a process called glycolysis. And these kind of like hypotheses, so that like neurons really consume majority of glucose in the brain and these metabolic profiles have been mainly studied in animal studies or in vitro, but haven't been really shown in humans on a whole brain level. And this already brings me to our aims. So the first aim would be like, can we really validate these profiles of neurons and astrocytes in the human brain on a whole brain level? And further, we were wondering if there's like regional heterogeneity of cell types and metabolic pathways. And lastly, we wanted to answer the question if like really neurons are the cells that consume majority of glucose in our brains. And the first two questions I would like to answer with transcriptomics and the last one with imaging transcriptomics. So let's start with the first question about like validating these metabolic profiles. So you might be wondering like why we use transcriptomics. So basically transcriptomics allows you to measure really how high like a certain gene is expressed. And what makes really a neuron, or what characterizes like a specific metabolic pathway is really the expression of all these kind of like specific genes. And if we know like how high or low a certain gene is expressed in a region, then we kind of have also like an estimate of like the abundance of neurons or like a metabolic pathway. And the Allen Human Brain Atlas has actually exactly this kind of data. So it contains transcriptomic data of 6 postmortem brains, and by the use of ABAgene, we can average and normalize this data and get it into our parcellation of choice. So then what we get in the end is like really normalized expressions of over like 16,000 genes. And if we then look at the genes that are like, for example, specific to neurons or like oxidative phosphorylation and take a Roy-Weiss median of these genes, we kind of get like an estimate of neuronal abundance across the cortex, or the same would be true for the metabolic pathway. And our general hypothesis is that like if really neurons are oxidative and a region is high in neuronal gene expression, then in turn also this region should have high expression of oxidative phosphorylation genes. Likewise, if like a region is high in astrocyte genes, it should also have like a high expression of glycolysis genes. So I generate these brain maps and basically correlate the cell types with the metabolic pathways. And indeed, I see that neurons really correlate with the expression of oxidative phosphorylation genes, while astrocytes correlate with the expression of glycolysis genes. So our first aim, we managed to validate really these two profiles of astrocytes and neurons. And on like a little side note, I don't do it only on neurons and astrocytes, but actually for all major cell types in the human brain. And I find that especially excitatory neurons are the cells that really rely on this high oxidative glucose metabolism, while glia cells seem to use other substrates such as fatty acids. And this works very well in cortical regions, while in subcortical regions we have like a different pattern. So for neurons, we don't really see a relation anymore with the metabolic pathways, but mainly for glia cells. Okay, so now to the second question, if there's like regional heterogeneity of cell types and metabolic pathways. So generally we can see that neurons are like higher expressed than astrocytes, and they seem to be like kind of stable across like different cortices. And in terms of the metabolic pathways, we see that glycolysis is kind of like also more stable and has like a bit of a gradient from posterior to anterior, while oxidative phosphorylation has this like very specific spatial pattern. And generally what we also observe is that really neurons have like higher expression in cortical regions, while astrocytes are higher expressed in the subcortex. And along with the higher expression of neurons in cortex, we also have higher expression of oxidative phosphorylation in the cortex. Glycolysis seems to be kind of more stable, so we have like similar expression in cortex and subcortex. Again, as like a little side note, I actually see generally that glia cells are higher expressed in subcortex, along with like glia-related pathways. Okay, so while it was very helpful for us to average like neuronal genes or like astrocyte genes to get like these metabolic profiles, when we average all these genes, we also lose like a lot of kind of information. Because as you know, like it's not only that we have neurons and astrocytes, but many different subtypes of these two cells. So what we were wondering now is like, do we see actually some genes that have like a specific spatial expression pattern across the cortex? So here what we do is we order the regions according to their position on the posterior and anterior axis, like as a proxy of, as an evolutionary proxy. And then we take all these genes here and run a clustering analysis. And what we actually find is that there's like two clusters, and one is like ranging from or increasing from posterior to anterior region, and the other one the other way around. And what is really interesting is that the cluster that increases in frontal regions is actually the cluster that contains mainly genes that correspond to astrocytes. And while the other cluster that is higher in posterior regions is the one that is higher in neuronal genes. And this really poses like interesting questions on what the function really is of glia cells, or in this case astrocytes, in frontal regions and also in higher cognitive functions. So I hope I could also show you that there is regional heterogeneity of both cell types and metabolic pathways. And now I want to answer the last question, if there's if neurons are really the cells that consume majority of our glucose. So we have these CMR glucose maps, and I get that. So that measures really the glucose consumption in the brain, and I get it into the same parcellation as my expression maps. And when I do the correlation, I actually do see that the neuronal genes, they correlate with our CMR glucose measurements, while the astrocytes do not. So generally, we can also confirm the last statement. So it does really seem that neurons are responsible for the majority of glucose uptake in our brains. As a take-home message, I really want to emphasize that the Allen Human Brain Atlas seems to be really highly reliable, and we can really do studies also on a cellular level. Firstly, next I would want to say also that we do see differences of regional differences of cellular and metabolic pathways, and also this kind of cell distribution across this posterior anterior gradient, or also as proxy of evolutionary gradient. And last, we do really see that neurons are the cells that consume majority of our glucose, and that our FDG PET signal likely reflects the uptake of glucose in neurons. And with that, I really want to thank the Neuroenergetics Lab, Valentin Riedel, my supervisor, and also my colleagues, especially Magnus, Roman, and Gabriel, who also helped with the data and the analysis, the funding. And also, unfortunately, my poster session is already over, but I have the e-poster, so feel free to check it out. And I'm happy to discuss it whenever you feel like it. And now, thank you very much for your attention, and I'm happy to take questions.
Thanks. That was a really compelling and great talk. I was just wondering, so you have this distribution of neurons. I saw it was kind of changing pretty dramatically in the subcortex. Just wondering if sort of the neuronal map that you're using maybe is based on cortical neurons, and maybe the subcortical neurons might be a lot more variable, and perhaps, is there a way to basically look at different types of neurons and see if that would change the sort of distribution you're looking at in the subcortex?
Yeah, that is true. Like, I think, like, if you have these, because these cell types marker, they actually originate from single cell studies, and majority are really from cortical samples. And we do also want to work now in the future with like a lab that really does single cell also, so to really differentiate the different kind of like subtypes. Because as we can see also in this like clustering analysis, there is really different kind of like, it seems like there's different subtypes, and if I average all these genes, it's kind of like I lose this kind of information. And I think to generally explain or like show these like metabolic profiles, it was helpful, but it's definitely worth to go more in detail about the different subtypes, yeah.
Cool, thank you.
Thank you.
So this is the last question.
Part of the brain was definitely missing in your diagrams, which was the cerebellum, which I think of as being somewhat unique in terms of some of its cell types, especially interneurons, that I think of as being very metabolically active. So I was wondering if you had any insight.
Yeah, I haven't included it. Actually, my first analysis was really cortical only, and then I started to extend it to subcortical regions. But I really would like to extend it also to cerebellum and also to white matter, actually, to verify. Yeah.
Thank you, everyone. Thank you again, Laura. Just as a quick note, if you still have questions, you can post them on the app, and we will give them to the speakers afterwards.